Hello friends, welcome to our YouTube channel. Today we will be discussing about the vulva formation in C. elegans because this topic is very important and lot of the questions has already been asked previously. So let us take a look toward this topic, and that is the vulva formation in C. elegans. C. elegans means Caenorhabditis elegans, and this is a nematode which is having 1 mm of length. Okay, and the most important thing is that it has a embryonic developmental phase of 16 hours, and uh, the cell which is dividing can be seen from the upper cuticle very easily, because the upper cuticle is very transparent and it can be seen easily. Dividing cell can be seen easily, and due to its low number of the cell, because almost you can say near about thousands, it is made up of the 1,000 cells, so it is very important, and it becomes very easy to trace all the cell division and activity undergoing inside the cell. So this is the reason why the C. elegans is most important model organism to study about the molecular biology and the developmental biology. So if you are talking about vulva, what is vulva? It's a ventral opening and through which the fertilized egg after division is expelled out for the development. Okay, so let us take a look on the figure which I have made it previously. So if you are looking toward the figure, it is very clear that this is a C. elegans, and whatever we have highlighted, that these two things, because we are going to study about only these two or three things. So this is a somatic gonads. Okay, this is a somatic gonads, and this is an anchor cell. Anchor cell, and this anchor cell will be responsible for radiating some sort of protein, and under the influence of this protein the fate of some different cell is decided. Okay, now these are the vulva precursor cells. Vulva precursor cells VPCs. Okay, so in coming lecture we will be discussing about the formation of the anchor cells, but in this section let us understand what is the exact function of the anchor cells. Okay, in this lecture we will be studying about the function of the anchor cell, and in the coming lecture we will try to understand how the origin of the anchor cell occurred. Okay, just whatever you see here, the ventral region. Okay, so in the ventral region there are some cells. Okay, and actually these cells are 6 in number. These cells are 6 in number. Actually what happens, these ventral cells are 11 in number. Okay, this ventral cell, or you can say the posterior cells, are 11 in numbers, because it is located ventrally and it is posterior part. So actually this posterior cells is you can say here for understanding anterior and posterior. Okay, so this ventral posterior cells actually having 11 in number. Okay, and this 11 in number can be categorized as PNP. Okay, PNP means P posterior. Actually the first P1 is the PE stands for the you can say the stem cell, N means number, and P means posterior. Okay, so from P1, P2, P11, P. So this is you can say the posterior epidermal cell, ventral posterior epidermal cell, ventral posterior epidermal cell. and out of 11, only 6 are selected is a vulva precursor cell. Okay, so till now we have understood about this figure. Okay, and let us proceed toward another figure. So when we take a section of this region only, we will get somewhat this structure. Okay, so this is structure. So already we have discussed about this structure, that this is gonads, somatic gonad. This is actually the somatic gonads that we have already written, somatic gonads. And now this is the anchor cell. Okay, this is anchor cell, and these 6 cells, if the 6 cell, actually the cell starts from P1, P2, P11, P. Okay, so what actually the scene is that the P3P, P3P, P4P, P5P, P6P, P7P and P8P. These 6 cells from P3 to P8, these 6 cells are recruited as the vulva precursor cells. Okay, and this is the anchor cell. Okay, so these 6 cells would be responsible for the formation of, or you can say here, the whatever the factors which is coming from the protein which is coming from the anchor cells will show the equivalence effect. Okay, so let us understand about the equivalence effect. So this thing will be after division. After division, the structure will be somewhat like this. So before proceeding, let us understand here, this is an anchor cell, and this anchor cell is responsible for the secretion of lin-3 protein. Okay, the secretion of lin-3 protein. Now what happens? Lin-3 protein undergoes, whenever it interacts with, and this is the late 23. This is a receptor, and the lin-3 will be emitted from, the lin-3 protein will be emitted from the anchor cells through, you can see the WNT pathway. And whenever this lin-3 protein, actually whenever this lin-3 protein this lin-3 protein this lin-3 protein will be taken up by the late 23 receptor. Okay, now what happens? This lin-3 protein is surrounded all where? This lin-3 protein is surrounded all where? This lin-3 protein is emitted and it will be taken up by every cell, P3P, P4P, P5P, P6P, P7P and P8P. Okay, so this protein is being released from the anchor cell, and it is being, there is no bias of the anchor cell that it will provide the lin-3 protein to a specific cell. So here the equivalence is, the law of equivalence is here, means for anchor cells all the 6 cells are same, but here what happens, those cell which is just in front of or which is in close proximity with the anchor cell will lead to the formation of the vulva. So if we see the figure, we find out that the maximum lin-3 is taken up, the maximum lin-3 protein is taken up by the P6P cell. Okay, so it is being taken up by the P6P cell. Okay, and it is taken up by P7 and P5 cell less than the P6P, means maximum number of the maximum number of the lin 3 protein is being taken by the P6P, and then after by P7P and P5P. Okay, and then after it is very less taken by P8P and P4P and P3P. So if we count that, if we say that the ability to form the bulbar cells will be highest in the P6P, means actually the P6P will be responsible for the primary vulva formation. It will be responsible for the primary vulva formation. P7P and P5P will be responsible for the secondary vulva formation. Secondary lateral vulva, you can say 1 degree, 2 degree, and the fate of P4 and P3 is 3 degree. Means you can say the 1 degree means P6P. P6P will lead to the central vulva formation. Okay, P6P. Then P5P and P7P, P5P and P7P will be responsible for the lateral vulva. And then after P8P and P4 and P3 here will be responsible for the formation of hypodermis. Okay, so this one will be the primary, means you can say vulva, a lateral vulva. This one is the hypodermis. This will form hypodermis, and this one is also, this one is the lateral vulva, and this one hypodermis. Okay, now here another factor works, and that is lin, which is the result of the notch signaling, produced from the P6P, and another protein is there, and that is called as the lin 23, lin 12, lin 12, lin 12, lin 12, lin 12, means all those cells which are responsible for, and all those cells which are getting the lin 3 protein will make another protein that is called as the lin 12, and this lin 12 protein will be effused laterally, and this will stop the other cells to make its fate. Means actually what happens, so first let us understand here, this lin 3 protein was taken by the maximally taken by what, that is called as the P6P, and this P6P will produce the lin 12 protein, and it will be radiated from, it will be taken up by the lateral adjacent cell, and it will act on this adjacent cell laterally, and this lin 12 here lin 12 will stop the adjacent cell to acquire the 1 degree vulva, or you can say to acquire the primary or central vulva formation. Means lin 12 will stop this lateral P7P and P7P and P5P to produce the central vulva. Okay, same here, this protein, this cell is also getting, suppose P7P and P5P is also getting the lesser amount of the lin 3 protein, and it will also radiate, or it will also produce the lin 12 protein, and it will also act on its adjacent cell. It will also act on its adjacent cell. For here, for this cell, this will be the adjacent cell, it means P4P will be adjacent cell, and P6P will be also then adjacent cell. Okay, so what actually happens, this P6P will affect this one, means it will affect the P4P cell not to acquire the lateral vulva fate, means not to acquire the secondary vulva cells. Okay, simultaneously it will also radiate, it will also give the Lin12 protein to P6P, and it will signal not to acquire. To whom this will radiate the signal, and this will affect this protein not to acquire the secondary vulva fat. So in this way, this protein, this Lin12 protein will act laterally to its adjacent cell to not to acquire its own cell fat. Means it will stop the P5P and P7P to acquire the primary fat of the vulva. Again, P7P and P5P will affect, or you can say will stop its lateral cell, that is P4P, not to acquire the fat of secondary vulva cells, and this will also radiate, or it will also influence this adjacent cell P8P not to acquire secondary fat. Okay, and in this way it will be secreted by the cells, and it will stop to form its own fat. So this was all about the vulva formation in C. elegans. Hope you like the video. If you like it, please don't forget to subscribe, share and like the video. Thanks for watching.